I'm Kurt Thorn, and today I'm going to be talking about two-photon microscopy, which is an optical sectioning technique that's particularly good for imaging thick tissues. In my previous lecture, we talked about confocal microscopy. Two-photon microscopy can be thought of as a variant on confocal microscopy that works for even thicker specimens than confocal does. So to start, I just want to review how ordinary epifluorescence microscopes work, how confocal microscopes work, and then I'll talk about the shortcomings of those that two-photon microscopy is trying to address. So if you remember from my previous lecture, in an ordinary epifluorescence microscope, we have excitation light that excites our sample here, this little blue object at the bottom. That's focused by an objective lens into the sample, and then the sample emits fluorescent light that is collected by the objective and projected by the tube lens on a camera here. So that gives you an image of the fluorescence from the sample, as shown by this green point here. However, you can also have out-of-focus fluorescent light that comes from other regions in the sample, say this region up here, shown by this green point, and that light is not going to be focused onto the camera. It's going to show up as a blurry object superimposed on our in-focus light. And so this will reduce our ability to resolve in-focus objects in the camera. So this is a general problem for epifluorescence microscopy, is that fluorescence is emitted along the entire illuminated cone in the sample, not just from the focus in the sample. So one way to get around this is to use a confocal microscope, which I described in a previous lecture. And here, we use a pinhole to block out of focus light. So the idea here is we, again, have our sample illuminated by this cone of light. It emits light from the in-focus point. And now, instead of a camera, we put a pinhole at the detection plane. And so that pinhole blocks out of focus light and passes the in-focus light. So you can see here the in-focus light passes through the pinhole and arrives at a detector to be imaged, whereas light coming from this other focal point in the sample that's out of focus hits the sides of the pinhole and doesn't pass through and therefore is not detected by the detector. And so this confocal microscopy uses this pinhole to block out of focus light and so give us an in-focus image of just a single optical section. So what limits the imaging depth in this case? What prevents us from using confocal microscopy to image arbitrarily thick specimens? So if we just consider the excitation path here, just looking at this excitation light going through the objective into the sample, there are two things that limit the imaging depth. One is a sort of trivial issue, which is the objective working distance. That this objective here sits some distance from the sample, and to image deeper into the sample, we need to move the objective closer to the sample. And eventually, the objective is going to collide with the top of the sample, and then we won't be able to go any deeper. So in a sense, that's a real issue, but it's also an issue that can be addressed by engineering. And you can buy objectives with very long working distances that allow you to image in several millimeters. There's another problem that limits imaging, which is the ability of the light to penetrate into the tissue. And in a way, this is something you're probably all familiar with already, that if you look at your hand, say, it's not transparent. And so that limits your ability to image into things, is that you just can't get the light in or out. So what limits that tissue penetration depth? So there are two issues here. So let's first think about the ideal case. So I've just drawn schematically here a tissue. Here's some cells, some deeper tissue, and then these red lines here are meant to represent the light being focused into this tissue to excite our fluorescence. And so if everything is perfect, this is what you'd get. You'd have your light coming in uniformly, it's not disturbed by the tissue, and it reaches a focus inside the tissue wherever we want to image. Unfortunately, in the real world, this isn't what happens. Instead, what you see is that some light will behave ideally. It won't get interfered with by the tissue, but some of the light that's focused in here will be absorbed, so it'll hit molecules in the sample that absorb the light, and so it just is lost. And other rays that are entering the sample will be scattered, so they'll bounce off objects in the tissue, off refractive index discontinuities, and they'll be scattered to new directions so that they no longer converge at this focus. And both of these effects are dependent on how thick the tissue is, right? Because there's, in each case, a constant probability per unit depth of being scattered or absorbed. And so as you go deeper and deeper, there's more and more chance that that light will be scattered or absorbed. And so you get less and less light reaching the focus as you go deeper and deeper into the tissue. And this is the really fundamental limit for imaging depth in real tissues in, say, human tissue or mouse tissue or other thick biological specimens, that both absorption and scattering will prevent light from being focused to a nice tight focus where we can then detect the emitted light from. So let's talk about absorption a little bit. Absorption can arise from many, many different molecules in the sample, and this spectra here just shows a couple of them. So this goes from the UV down here at the left all the way out to the far infrared on the right. And we're looking at a couple of different molecules here. In black, this black curve is water. And so as we all know, water is very transparent in the visible. It's clear. And then proteins in samples absorb in the UV. There are molecules like hemoglobin, shown here in these green curves, that absorb. Melanin, the stuff that makes your skin dark, absorbs. And then as you go into the infrared, water starts to absorb, and then you get molecules like collagen here in blue absorbing. And the sort of takeaway point from this side is that the visible is pretty bad, and things get better as you go into the red and the infrared. And this explains sort of the common trick you've probably done as a kid, where you shine a flashlight up to your hand or your cheek, and you see red light comes through your skin, but not the green or the blue light. And that's because these absorptions all drop off here in the red, and they get even better as we go into the near infrared to wavelengths around 1 micron. So you can see there's this sort of window here that's been indicated by these blue arrows, which is a particularly good place to image in biological samples, which is the far red and near infrared. And that's good because none of these molecules absorb there, or they absorb very weakly. So let's talk a little bit about scattering. Scattering also is wavelength dependent. One of the dominant sources is Rayleigh scattering, where the amount of light scattered goes as 1 over the wavelength to the 4th power. So this also gets better in the infrared. As we go to longer wavelengths, there's less scattering. And as we go to shorter wavelengths, there's more scattering. So this also suggests that we should be imaging in the near infrared. And so as a result, imaging in the near infrared, the far red to the near infrared window, this window around 1 micron, is really good for imaging into thick biological tissues because it minimizes absorption and it also reduces scattering. Before I continue talking about infrared imaging, I just want to make a little tangent here, which is to discuss other approaches of getting rid of this absorption and scattering, and that is this mechanism of clearing. And this is a little bit of a vague term. It's been used to apply to many different processes. But in general, this is the idea that you can take your tissue sample, here these are mouse embryos, and you can soak them in some chemical solution that will dissolve out the scattering materials and the absorbing materials, and also permeate the specimen and equalize these refractive index differences that can give rise to scattering. And so this is a recent paper from the Miyawaki lab, where they developed a particularly good chemical compound for doing this called scale. And shown on the left here is a mouse embryo that hasn't been treated. And then on the right here is a mouse embryo that has been treated. It's been soaked in the solution for a couple of weeks. And you can see that now you can see through it. It gets very clear and transparent. And you can see this pattern on the paper behind the embryo through it, which you can't see through the uncleared embryo. And so this is one approach to deal with this problem, is to treat your samples to eliminate these problems. And that gives rise to very deep imaging. So here's a 2 millimeter image into a mouse brain in one of these cleared samples from their paper. The downside, of course, to this is it doesn't work for live samples. You can't take a live mouse and dissolve out all this stuff and still have a live mouse at the other side. So this is one approach that can be used and is very good for fixed samples, but still doesn't really solve our problem if we want to be able to image into a live animal. So the absorption and scattering concerns I've discussed suggest that really we should be imaging in the infrared. And there's a couple possibilities to do that. One is to use infrared excited dyes. So these are dyes like Ti-7 or other dyes that are excited at 700, 800 nanometers and emit around 900 or 1,000 nanometers. These are commonly used for whole animal imaging, these kinds of devices where you are looking at tumors, say in an entire mouse. You take an anesthetized mouse, inject it with one of these dyes that will bind to a tumor, and then you put it in a device where you just look through the skin of the mouse at the whole animal. And these work very well for that approach. The other approach, and the one I want to talk about today in detail, is an idea that you can use 2 photons to do the work of 1. And so you could take a dye that's normally excited in the green, say, and now image using 2 photons at twice the wavelength, so 2 infrared photons, and use that to excite this green dye. And now you can use your standard dyes, but excite them in the infrared. So to explain how this works, I'm going to take a step back here and talk briefly about conventional excitation in microscopy. And Nico, I believe, has a lecture on fluorescence microscopy that explains some of this. And the idea here is that there is, your dye has 2 energy levels, the ground state here, this S0 state, and the excited state, S1. And in normal fluorescence microscopy, you use a wavelength of light, here we're showing it as blue, that is high enough energy to excite this molecule from the ground state to the excited state. There's some internal conversion here where this relaxes. And then eventually it drops back down to the ground state, giving off some emitted light, these green photons here. And this is conventional, one-photon fluorescence microscopy. Now, the idea for this infrared imaging using 2 photons, two-photon microscopy, is that if you use 2 photons that are half the energy of that blue photon, if they arrive at the same time, they can add together to make up essentially 1 blue photon. So that you absorb essentially at the same time these 2 red photons that each have half the energy of the blue photon, and that's enough to take you from the ground state to the excited state. And now you're back where you started, and you can relax down to the ground state and give off these green photons again. And this works. The trick to making it work is that the photons have to arrive nearly simultaneously within a femtosecond, so 10 to the minus 15th of a second. So essentially, instantaneously at the sample, these 2 photons have to arrive, be absorbed, and then that will promote the molecule to this excited state where it can then fluoresce. And this idea is called 2-photon excitation, or more generically, multi-photon excitation. Because we require these 2 photons to be absorbed nearly simultaneously in the sample, this means that the brightness of our sample, the fluorescence intensity, is going to depend on the square of the number of photons arriving there, because we have to not absorb 1, but 2. So we have to multiply the probability of absorbing the first photon times the probability of absorbing the second photon, and that gives rise to this dependence on the square of the excitation intensity. And this turns out to be actually a very nice property of 2-photon excitation, because it means that we have a nonlinearity in our excitation properties, which gives rise to a localized excitation. And that's demonstrated here in this fairly famous photo from Brad Amos at Cambridge, where we have 2 objectives shining light into a cuvette full of dye here. This objective on the left here is shining in the green light, or the blue light that gives rise to the 1-photon excitation. And you can see there that this light is absorbed throughout the entire path that the laser beam travels through the sample, because everywhere there's light, there's going to be absorption. There's just the amount of absorption is linearly proportional to the intensity of the light. This objective on the right here, however, is now shining in light that's twice that wavelength. It's at an infrared beam that requires 2-photon absorption to give rise to fluorescence. And here now you see that basically the only place you see fluorescence is this tiny little speck in the center there. And that's because that's where the intensity is highest. That's the focus of that beam. And so it's the only place where the laser intensity is strong enough to give rise to that near simultaneous absorption of 2 photons. So because we depend on the square of the excitation intensity, instead of seeing this hollow cone of light illuminating our sample, we get only excitation from the tight focus where it's most intense. That can also be seen in this slightly more elegant image here from UC Berkeley, where you've got a microscope objective again shining into a cuvette. Here's the 1-photon case where you see this entire cone of illumination. And then here's the 2-photon case where, again, you see this little tiny speck, and there is a zoom in here right there. And so this is the real strength of 2-photon excitation, is that there's no out-of-focus light. We're only getting excitation in the region where the beam is most intense and most tightly focused. So we only get excitation from the focus of the beam. So the consequence of this is that we essentially get confocality for free. And let me explain that. So the idea is in confocal, the focal volume, the region you detect on your detector, is defined by the point of light. So it's defined by the focus of your laser beam times the detection pinhole. So you have a point excitation times a point detection, and that gives rise to detecting only the stuff that's in focus as one point of excitation. In two-photon excitation, the focal volume is defined by the point of light times itself, because we need the square excitation. So you're multiplying that excitation cone times itself. And so the net effect is you get the same point spread function as confocal, but you no longer need the pinhole. So we've now really simplified our optical system. We can get rid of that pinhole and still get the same resolution and the same Z-sectioning capability. So let's go back and see how this applies to a case where we have absorption and scattering. So again, here's our tissue. Here's our focused beam going into it, and bad things are happening. And there's light being lost by absorption and scattering, but so long as we have some light coming to a focus here, if we turn up the brightness high enough, if we turn up the intensity high enough, we'll get enough photons here to do two-photon excitation. And so we'll just excite this little green dot here at the focus of that beam. And now, unlike our one-photon case where these beams would all be giving rise to excitation of the sample throughout the tissue, we only have excitation at the focus of the beams that do make it to a point. And that means that we know that all the emitted light comes from that focus. And so one consequence of that is that it doesn't really matter now what happens to the light coming from this focus, whether it comes out in straight beams or if it comes out and is scattered. We can have terrible scattering here. We can have this green light bouncing all around, and it doesn't really matter because we don't need to image it. We just need to record the intensity because we know that any green photons that come out of this had to come from this focus because there's no other points being excited in here. So that, the fact that this one comes out straight, but every other beam is bouncing off of stuff in the tissue and coming out at strange angles, doesn't matter because we don't need to image them. We just need to collect it and record how bright it was. And so this is the real strength of two-photon microscopy and what makes it so useful, is that we can eliminate the pinhole from our optics, and we don't need to image anymore. We just take essentially a bucket and collect all the light that comes out of our sample in it, and we know that that came from that one focal point. And so the microscopy manufacturers call this non-descan detection. This is a little bit of a sort of a technical term, but it essentially means that you're not using a pinhole. So in a regular confocal microscope, you descan the light, you remove the scanning from it so you can pass it back through a fixed pinhole. In two-photon, you can eliminate all those optics, just collect the light and count how intense it was, and you know it came from that point. And so if we do this, then this is basically now the same process as confocal microscopy. We raster scan our beam across the sample. So we take our infrared beam, focus to this tight point, scan it across our sample here, so we make this grid of pixels. And at every pixel, we record how much of the emitted light comes out of the sample. And that gives rise to our final image here. And so that part of the microscope is, the excitation part is really identical to a confocal. And then the detection path is this much simpler path where we don't need the pinhole, we don't need to descan the beam, we just record the total emitted light. And so this is called, as I said, non-descan detection. And you want to use that for two-photon because if you can do two-photon in a conventional confocal and keep the pinhole in the detection path. But then you lose all this light that doesn't come to a tight focus at your pinhole, and so you get much, much less sensitivity. So eliminating those optics is really the way to go if you're trying to do two-photon microscopy. And if you're not doing that, you're essentially missing the real strength of it, which is that you can eliminate this stuff and collect light that would otherwise not be properly imaged to your sample. So this can be pretty simple to do. This is a picture of a home-built two-photon microscope from Max Krummel at UCSF. And what you see here, so this is the laser beam path. It comes in through here. There are these scanning mirrors here. These are the mirrors that raster that beam across the sample. So those are the ones that will let you scan that spot pixel by pixel across the sample. Then it's a little hard to see here, but there's a filter cube in here that directs your beam down to the sample, which sits on the stage down here. So the objective is totally hidden under the microscope here. Light from the sample comes back up and then is detected under this array of PMTs here. So these are the photomultiplier tubes that record your light from the sample. And these are basically identical or very similar to the photomultiplier tubes used in conventional confocal microscopy. Just again, you don't have the pinhole in this path. So this allows you to do detection of multiple channels simultaneously. This is just showing here a scheme for splitting different wavelengths onto different PMTs so you can record multiple channels at the same time. And I'll come back to this a little bit later. And so that's the basics of the two-photon microscope. Now I want to talk a little bit about how we get the excitation here, and in particular the kinds of lasers we need to use for doing two-photon excitation. So as I said, the brightness of our sample, the emission intensity, depends on the square of the excitation intensity. So that means we need a very bright excitation. We need high excitation power. And the way we do that is to use a pulsed laser to get high peak power so that we get a lot of photons in a very short burst, but without using tremendous amounts of power into our sample at all times. So the idea here is in a typical laser, a so-called CW or continuous wave laser, we have constant intensity per unit time. It's like a light bulb. It's just on and it shines light at some intensity however long it's on. In a pulsed laser, what we have instead is these pulses that are emitted by the laser. So now the laser is dark most of the time, and then only in these short bursts does it emit light. And so a sort of typical laser you would use for two-photon microscopy, this is called a Ti-Sapphire laser. It's a titanium-doped sapphire crystal. And the particular one I'm giving the specs for here is this Ti laser from Newport. These pulses are 80 femtoseconds wide. So these are very, very short pulses. They last, a tiny, tiny fraction of a second, 1 part in 10 to the minus 14th of a second. And there's actually quite a lot of them. They are repeated at an 80 megahertz rate. So there's 80 million pulses per second, and that means the time between pulses is 12.5 nanoseconds. So this laser is mostly off, and then every now and then it comes on and it has this very bright burst of light. And so the idea here is it's only on about 150,000th of the time. So it's off almost all the time, and then it comes on very briefly. And so that means if we average the power over this, so the average power consumption of this laser can be something fairly reasonable, like 1 watt, but that means that the peak power here is 150,000 watts in one of these pulses. So it's an incredible amount of power. There's no way you could ever run a continuous laser at 150,000 watts. You'd need a small power plant, to run your microscope. But these things, these pulse lasers get you that very high peak intensities, which you need to guarantee that there'll be 2 photons arriving at the sample at the same time, while maintaining a sensible average power that's not going to require huge amounts of electricity or burn tremendous holes in your sample. The downside to these things is they're not cheap. They run you sort of $100,000 to $200,000 just for the laser. And so this means that a two-photon microscope isn't a cheap microscope, but it lets you do things that you can't really do any other way. One other nice thing about these lasers is they're tunable. So by just adjusting the position of optics inside the laser, you can change the emission wavelength over a range of about 700 to 1,000 nanometers. And if you remember back to the slide I showed of the absorption of biological molecules, this is exactly the range you want to work into to minimize that absorption. And this is why these titanium sapphire lasers have become the really popular workhorses of two-photon microscopy. They have this very nice pulsed behavior, and they have this tuning range that exactly matches what we want for biology. So now let me turn to talking a little bit about the dyes and two-photon absorption and two-photon excitation in real dyes. This is a figure I took from a recent paper looking at two-photon excitation of fluorescent molecules, fluorescent proteins, and in particular these are red fluorescent proteins of various kinds. And what's plotted here is you can see in these black curves the excitation spectrum, the one-photon excitation spectrum for these 9 different molecules. And in these red dots are the two-photon excitation spectra. And these are plotted in such a way that the one-photon excitation wavelengths are on the top grid here, and then the two-photon wavelengths are on the bottom scale here, and the two-photon wavelengths are plotted exactly at double the one-photon wavelengths. So if the two-photon spectrum was exactly twice the one-photon spectra, these red dots would overlie exactly on the black curves. And you see that they don't, and in general you can see, for instance, for this guy down here, they're shifted to the shorter wavelength side. So you can see this red peak here is shifted to a shorter wavelength than the black peak. Same for this guy here. There's a wavelength shift. And this is what's generally observed, is that the two-photon excitation spectrum is not exactly twice the one-photon excitation spectrum, and often it's shifted to shorter wavelengths. Generally, you just measure these things, or you go to papers like this to figure out the best way to excite your molecules. Here's another example. These are synthetic small molecule dyes from Invitrogen. And these are dyes that absorb at a wide range of one-photon wavelengths from 350 here to 594. And you can see that many of them sort of have fairly similar excitation maxima. And you can also see that these spectra are fairly broad, that all these dyes can be excited to a large extent over 100 to 150 nanometer range in wavelengths. And this means that you can use one excitation wavelength from your laser to excite multiple dyes. And this is both good and bad. It means you can't pick out a single dye by changing your excitation, but since tuning these lasers takes some time, it's not instantaneous, it also means you can excite multiple dyes at a single wavelength of your laser, which means it's fast to acquire multicolored data. And so that brings us back to this detection path here, where we have these multiple PMTs split up by wavelength. And so you see here's the emission comes out of this laser, and then it's just split by these different dichroic mirrors into different wavelength bands. And so the idea here is you take advantage of the fact that one wavelength will excite these many dyes, and then you can record the emission of these multiple dyes in these different channels basically simultaneously. There's another mechanism that can give rise to two-photon excitation that I want to talk about, and that's called second harmonic generation. And this is not a fluorescence mechanism, but it's a mechanism by which an anisotropic molecule can frequency double the light. So it can absorb a photon at, say, 1,000 nanometers, or absorb two photons at 1,000 nanometers, and then emit a single photon at 500 nanometers. So it exactly doubles the wavelength, or doubles the frequency of the light, halves the wavelength, doubles the energy of it. So it's a mechanism of converting two photons into one at twice the energy. And this has similar properties to the fluorescence in that it scales as the square of the excitation, but only a very few things do it because of this requirement for anisotropic molecules. So it tends to be structural molecules, things like collagens, only some kinds of collagens actually, or cellulose give rise to this. And so this can be very handy if you're imaging tissue. You see here's I think this is a mouse lymph node, and you can see this meshwork of collagen fibers overlaying it that were imaged by second harmonic generation. So if you're looking at something that has collagen in it, or you're looking at plant tissue with cellulose, you sort of get these things for free. And so you can pick out specifically that doubled wavelength and then image that to see where these structures are. This is also a nonlinear process, and so its brightness scales as the square of the excitation light intensity too. So that means you get the same sectioning properties with the second harmonic generation that you do for the two-photon fluorescence. So that's pretty much all I want to say about two-photon microscopy. I just want to conclude here by discussing a little bit when it's best to use it and when it's not so good. So two-photon microscopy's real strength is that it can image very thick specimens. So ordinary one-photon confocal at best can do about 200 micron thick specimens, whereas two-photon has no trouble doing 200 microns, and in fact it can image out to several millimeters thickness. So two-photon is great for imaging thick specimens, particularly if you need to get data from the entire thickness of this sample. It's also very good for live sample imaging, and that's partly because in fixed samples there are other ways you can solve these problems. As I mentioned, you can use these clearing solutions to dissolve away or otherwise remove that autofluorescence and absorption and scattering. But even more simply, you can often, in fixed samples, just cut sections. If you don't need to image the entire thickness of the brain, you just need to image some region of it, you just cut that region out and image it. Of course, again, that only works if your mice are dead or your samples are fixed. You can't do that in live samples. And so where two-photon microscopy really shines is when you need to image samples in live animals, particularly if you need high-resolution imaging, looking at single cells inside a live animal. There's really no other technique that can do this. And so that's the real strength of two-photon microscopy, is looking at cells migrating in lymph nodes. There's been a lot of immunology work done with two-photon, looking at T cells and B cells moving around in live animals. A lot of neuroscience looking at processes in the brain, imaging deep into the brain in live animals, things like that. So two-photon pretty much goes hand in hand with imaging live animals, either fish or mice or other objects that are significantly thick compared to what you can do with a one-photon or confocal or a spinning disk confocal. So I'll conclude there, and I just want to thank Max Krummel and Sebastian Peck at UCSF. Sebastian runs an imaging center called the BIDC that does a lot of two-photon microscopy, and Max is the director of that. And both of them provided some of the slides and quite a bit of information about two-photon microscopy to me while I was preparing this talk.